Okay. Can everybody hear me at the back? Yeah. Okay. It's a great pleasure to be back in Japan. Hiro and I know each other for at least 10 or 12 years, maybe 15. And so I would come here regularly, not to Tokyo, but to Okazaki. So it's a real great pleasure to be here. Also want to thank Michelle, who's somewhere from Sophion, for putting this great presentation together, and all the Sophion team here in Japan who've made this really a fantastic conference for everybody here. So my title's changed, but it's still really about AMPA receptors. And so I guess in summary, my lab is really interested in the building blocks of the nervous system. We're interested in different kinds of ion channels. So if you go to some of the posters that we have here, you can see that we're interested in these. We've had a long-held interest in ion-trophic-glutamate receptors. And it's in that context that we found ourselves interested in looking at a condition called intellectual disability, and that's the title of that. And it's also in the context of that I was actually contacted a few months ago by this magazine called The Scientist, which is quite an influential magazine in North America. And they asked me to write something about intellectual disability and where did I think the field was and everything like that. And so you get a chance to read the article. It's about 60 minutes or so to read it, or you can listen to the podcast. I basically argued that there should really be an intellectual disability research requires a unified strategy. And that sounds like a funny thing to say, but the reason I really said that was, first of all, for all the families, because I've got to know many of the families and the patients that are involved that have intellectual disability. And one of the big challenges in intellectual disability is that the origins, the genetic origins, which have been identified, are many, many fold. And so they have a hard time to try to convince politicians that they should come up with some common strategy in which how to go after that. And so if you read the article, you see I come up with some sort of suggestion for that. And then the other thing goes deep down. I'm a hardcore scientist. I wrote it for the scientists because if you've been following this space, then you'll see there's been a whole bunch of failures in different kind of drugs being brought forward for different cases of intellectual disability. I'm interested in Fragile X syndrome, amongst other things. And just recently phase 3 trials that failed on that. And I think part of the problem is that we really don't have a unified scientific explanation, sort of not just a political one, but a scientific explanation of that. So one of the challenges that the families, et cetera, that face is that politicians say that, well, these are rare or ultra rare disorders, these disorders of intellectual disability. But in fact, if you compare this 1 second, if you compare the prevalence of intellectual disability, about 1 to 3% of the population, and that actually, in terms of prevalence, is actually more people that are living on the planet right now with cancer. So it's actually a very prevalent disorder that's really underrepresented in terms of that. So before I get into the science, let me just quickly make a disclosure. So I'm actually the co-founder and CSO of a company called NOS Pharma, which is developing treatments for rare genetic neurological disorders, so things I'm very talking about. So why did I write this article making the argument that you really should be thinking about glutamatergic synapse as a real central hub for all of this? And it's kind of obvious. You're an undergraduate neuroscience student, and you realize that, in fact, ion-trophic glutamate receptor synapses are critical for brain development, learning, and memory. This is how we thought about it. If you can see my little laser here, this is how we thought about things about 30-odd years ago. But in the ensuing time, with cloning, with proteomics, electrophysiology, which I got involved in, we've identified a whole slew of different kinds of proteins, which are all disrupted in intellectual disability. And it turns out the epicenter of that is all around these glutamatergic synapses. And so although the different kinds of proteins, it turns out if you mess up that synapse in different kinds of ways, you end up with the same clinical features. So I teach medical students, and one of the striking things is when you look through the clinical profile of patients, regardless of the genetic origin of intellectual disability, they're all almost exactly the same. And that's also true for conditions like Fragile X, which misses FMRP. FMRP sits in glutamatergic synapses. SYNGAP1 as well is found to sit in glutamatergic synapses. And SHANK3, which is Phil McDermott, sits in glutamatergic synapses. You can even see it here as well. So the really disease of glutamatergic synapses, right? And I think if the industry and scientists start to think like that, then we might actually be able to get together and say, okay, what are the key areas that we should try and go to rather than trying to chase a whole bunch of other things? So two very important proteins that we've been interested in is the AMPA receptor, and that's going to be the subject of my talk today. And there's also a poster from Zhe in the lab. He's presenting some work on that. We're also interested in AMD receptors. And there's also a poster from Cecilia. I think it's tomorrow. She's presenting on that as well. Now, what's interesting about these two different types of glutamate receptors is that they're found in the same synapse, and they work kind of hand in hand. And the way we kind of thought about it is AMPA receptors kind of take the second role, the kind of subsidiary role where they depolarize cells. And the reason cells need to get depolarized because NMD receptors are chronically blocked by magnesium ions at resting membrane potentials. And the reason that's important is because when the AMPA receptor depolarizes the membrane potential, then the magnesium pops out of the NMD channel, transports a lot of calcium, and it's that calcium that's fundamental for learning and memory processes. So it's kind of obvious why you might think that disruption in NMD receptors might be tied into intellectual disability. You see with all these little kids, unfortunately. But it wasn't really obvious to us why deficits in AMPA receptors could be tied to intellectual disability. And so this data, which I put together from some of the stuff that we've got unpublished stuff, shows the structure of the AMPA receptor, which is this enormous protein that sits across the synapse. And then there's 4 different genes which encode GLUE 1, 2, 3, 4, and you can see the different prevalence of kids with intellectual disability with this. What's interesting is that if you look at the amino-terminal, the ligand binding domain, which is a region here, or the transmembrane region, so the pore-forming region, which you're learning about with all these ion channels, then you can see that the missense mutations are actually spread all across the protein. And that didn't really make a lot of sense to us because many of the missense mutations don't even disrupt the expression of these AMPA receptors. So what we considered is that there must be some new bit of biology that was going on with AMPA receptors, and here was kind of alluding to that, right? So what we decided to do is kind of go back and just study this in the process. So what we did is we recorded from a prototypical cell that had been studied before. So this is Purkinje cells here in the cerebellum, if anyone's familiar with that. And there's also these astrocytes in the cerebellum that literally sit right next to Purkinje cells. And these are glial cells here. Purkinje cells were well known to be calcium impermeable. I'm going to explain to you how we concluded that, whereas the Bergman glia were shown to be calcium permeable. It's generally thought, it was thought at the time, that most 95, 90%, 99% of all the AMPA receptors in the brain were all calcium impermeable. And the way we did these experiments is you record, you do these outside, these are not easy experiments, by the way. You pulled out outside of patches from these cells, you record it in sodium-rich solutions, you get a nice rest potential of 0 millivolts. Then you switch it into high calcium, and if it's very calcium permeable, the rest potential will be positive, and it's very calcium impermeable. You can see it's actually very negative. So Purkinje cells are not very calcium permeable. Bergman glial cells are very calcium permeable. What we found is that when you put them in solutions rich in sodium, you see they don't actually have a linear IV plot, and that's because they're blocked by polyamines, and that's something I identified a way back. I hate to say how long ago it was, but 95 with Mark Mayer, we had the neuron paper on that. But then when you replace all the sodium with calcium, you get this huge positive shift in calcium permeability shown in the Bergman glia for calcium. So we just established something that we'd seen before, and just to give you I did a shout out. This was done by this really talented PhD student who's now going on to bigger and better things, Ryan Alexander, who's going to be giving a talk. You guys are lucky. He's going to be giving a talk in the next session. So coming soon. So we started off looking at this problem by just thinking about why do we take the less frequently occurring AMPA receptors, the calcium permeable ones, and try to understand how do they transport calcium? It never really been solved. And we teamed up with this Japanese colleague who's actually at Vanderbilt, that Hiro I think knows, Teru Nakagawa, who's really an outstanding structural biologist. So we teamed up with him. And what we found is that when he was able to get the AMPA receptor into a very stable open configuration, which is easier said than done at this high resolution, he found, in fact, there was a calcium binding site that sat outside the membrane electric field. So it sat outside the pore region. The reason that was so important was that when you looked at the sequence alignment of all the different types of glutamate receptors, including the GluA2 containing AMPA receptors, then they also had that calcium binding site. So here's a little cartoon of the calcium binding site here. So all of the AMPA receptors, all glutamate receptor families have this calcium binding site. And so we said to ourselves, maybe it's not true that GluA2 containing AMPA receptors are calcium permeable. maybe they are, and everybody just overlooked it. And the reason we thought they may have overlooked it, so we thought maybe the glue took in AMPA receptor calcium permeable. The reason we thought they may have overlooked it is because in the last 5-10 years or so we found a whole series of different kinds of auxiliary proteins that bind to AMPA receptors. The most prevalent ones are these claudin family, specifically the TARPs, but there's also GSG1L, which we showed represents about 5% of all the AMPA receptors. And they're the kind of core part of a native AMPA receptor for the brain. And then they team up with cornichons, various other proteins. The other thing that's interesting is that these AMPA receptors actually are found in specific brain regions. So the TARP-only AMPA receptors are found in the cerebellum, whereas if you go to the forebrain, like the hippocampus and the cortex, then not only do they team up with the TARPs, but they also team up with cornichons. So there's maybe something very important about that TARP-cornichon thing that allows the hippocampus and the cortex to do such high levels of computation that are going on. So from that, we've actually developed a set of rules, which will come out in a review we've written for Nature Reviews Neuroscience. We sort of say that everybody already knows this, AMPA receptors, most of them are GluE2 containing, so therefore should be calcium impermeable. We're going to come to that. Most assemble with TARP, so that's proteomic work. Some are GSG1L, and then you get combinations of TARPs and cornichons in the hippocampus, and also with TARPs and CCAMP, and we get that. So how do we get to this? So the first thing we did is we built on some work that we had a neuron paper a bunch of years ago, showing that in the cerebellum, there were two kind of types of AMPA receptors. There was one, so here's the whole AMPA receptor here, and then they had two TARPs, two gamma-2s. There's a certain type of these TARPs are found here. However, the Purkinje cells actually had 4 TARPs on them. So what we said is, why don't we just simply recapitulate what we found before in the cerebellum in a recombinant system and just look at calcium permeability? Should have been something that you would imagine that people have done a long time ago, but they didn't. And so what we find is if you just take the pore-forming subunit, then they're not very calcium permeable. But if you start to add in these TARPs, you put 4 in, it's a little bit of a shift that's not really super compelling. But as you go all the way along, especially if TARPs are associated with the GluE2 subunit, which is a very dominant part of the gating process, then you actually get a pretty significant shift in calcium permeability, suggesting and kind of overriding what people have thought for the last 30 years, that AMPA receptors can't be calcium permeable. Turn your attention to the hippocampus, and here they form octamers. Remember I told you the TARPs and the cornichons there, so we tested them there. What we found is you get even bigger boost when TARPs and cornichons actually teamed up with each other. So here's the cornichon effect. So now it's actually transporting calcium much better than even sodium ions, right? So these are now very calcium permeable AMPA receptors that everybody has kind of overlooked when they've been looking at the work. And it was, again, certain kind of combinations, only certain kind of combinations worked for this. Now, when we put everything together, I'm not going to take you through all the data, but what we found, in fact, when we looked at all the various combinations, including native systems, we found there was a continuum in this calcium permeability. And that was really surprising even to us. But the other thing that was really striking is that if you looked at only the native forms of these AMPA receptors, it seems like there was a specific line. So the calcium couldn't be any more transported better than sodium ions. Because once it got beyond that, so when we found, I'm going to show you a picture in a minute, these kids, when we made mutations in the QR site, which in this site here, or in various other sites, that changed the calcium permeability of these receptors were found in kids with intellectual disability, but now they transported calcium much, much better. So we think that GluE2 containing AMPA receptors are calcium permeable, but there's a very strict limit. Once you go beyond that, it actually becomes detrimental to the brain. Okay? And so here's some of the kids that actually we studied their mismutation. This little girl here and this little boy here. And so what we found in her case, there was an exaggerated amount of calcium transport. And in his case, it was less amount of transport because it also affected the expression of this. And so as Hiro kind of alluded to, this came as a surprise to everybody. And I'm going to tell you in a minute why it really should have been a surprise to everybody. So much so that Nature wrote up a sort of review article, a sort of editorial that went with it, saying that, you know, this textbook assumption, which I had been teaching at McGill for the last 25 years or something, turns out one of the students emailed me, says, you're going to have to change all your lecture notes, right? So there's a kind of real change in how we think about these AMPA receptors. And a bunch of colleagues of mine, a bunch of friends who are now on faculty positions emailed me and said, oh my God, how did everybody miss this, right? And I said, well, actually, it's all of our fault, because if you know your evolution, then you'd realize that, in fact, that for the last 800 million years, AMPA receptors, or at least the ancestral AMPA receptors, were very, very calcium permeable. And this is just a little graphic to try to illustrate that here. So here's some non-bilaterian species up here. This is, I'll get into more complex organisms that become the insects. It's the protostomes, which are shown here. And here's the beginning of the vertebrate species. So if you just look at the AMPA receptors, you can see that the pore-forming subunits of AMPA receptors were around, at least 800 million years ago. And I'm going to show you a video of a trichoplex here, where it seems to be using an ancestral form of AMPA receptors. Same thing with auxiliary subunits. But what's interesting is that the RNA editing, which is important for this calcium permeability, and another thing, alternate splicing, which we also work on, they only turned up in early vertebrates. So in fact, AMPA receptors were never calcium impermeable. They're actually always very calcium permeable. And it was only with the occurrence of RNA editing that actually was attempted not to shut down the calcium, but just to make it more restricted, because the real estate around calcium in the brain is actually very, very important. So what about that in the context of very, very early organisms? So there's this organism called trichoplex, which is shown here, and it feeds on algae basically in the sea. This is a video of it feeding in a culture dish in this case. But what's interesting, we just did a paper with Adriano Sanatori, who's an evolutionary biologist at the University of Toronto. So he's kind of a couple of hours away from us. But what we found is that the ancestral AMPA receptors, this AKDF1, they're actually all found in these cells called these lipophil cells, which are involved in digestion, right? So the idea is that when this little organism here, maybe I can play it again, just to, maybe I can't, go back and play it again. But when it's feeding around, it's actually dumping out digestive juices. And it's thought that the algae release amino acids, and those amino acids actually activate these ancestral form of AMPA receptors. So it's as if the AMPA receptors that we have in our brain, which are critical for learning and memory, their original purpose, at least in these organisms, was tied into digestion, which is really interesting and unexpected, but not really if you think about the evolution of things. So in other words, AMPA receptors got kind of co-opted in early vertebrates into being used for fast synaptic transmission. Earlier on, doing something quite different, right? And the cytan-LAF, if you look at the sequence, the cytan-LAF is actually found in these trichoplex organisms. So cytan-LAF, this site G site that we identified in this Nature Structure paper, was something invented by biology, 800 years ago. Not 800 years ago, 800 million years ago, right? So it's an old invention in biology. So why do we care about that? So the reason we care about that, because we're really interested in human biology, is we haven't published this yet, but if you look at the pore region of AMPA receptors, so here are the 4 different genes, GOE1, GOE2, GOE3, 4. What you find is that if you look at the different kinds of missense mutations that have been found so far, and there's still more missense mutations being found in those kids. I just came back from Edinburgh in the UK, where they had the GRE, it's called the GRE UK Foundation. They had a conference last week to speak at that as well. There's been more of these missense mutations have been found. But you can see that the transport of calcium in the pore region seems to be a kind of epicenter in terms of disruption. And so we think that maybe disruption of calcium transport in AMPA receptors may be a fundamentally important, at least one of them. We think there's another thing that I want to tell you about, but at least one of the very important reasons why these kids end up having a perfectly normal brain, but yet they're unable to function as well in our society, is that during development, that calcium transport, which is highly regulated right inside neurons, is just disrupted enough that it kind of isn't enough to completely kill, but it's enough to completely disrupt their ability to kind of go through the usual intellectual processes that we all take for granted. And that idea is kind of getting some weight. And so I just noticed there was a recent Nature Review paper that was published by a bunch of clinicians. And they were speculating that based on other reasons that I'm not presenting, but other reasons that maybe disruption in calcium transport might be at the heart at least one of the things at the heart of neurodevelopmental disorders. So given that, I think it's a kind of rosy picture, 20 minutes, a rosy picture, because we now have something to try and go for. And it means if we can team up with drug industry and convince them that there's some tangible things we can go for, then we maybe stop wasting money on clinical trials that keep failing and disappointing these populations. So just before I finish, in the last 20 seconds, just want to thank all the people involved. It's not just my lab, but we team up a lot of people across the world for this. Here are two of the three first authors of that Nature paper. And I want to quickly introduce you to Je and Cecilia, who are embarrassed that I took their photograph this morning. Please go and see their posters. Ju's got this great poster on Fragile X, on AMPA receptors. He's got a new angle on it. And Cecilia's got this cool story about NMD receptors dysfunction, Fragile X. And she's got a pharmacological treatment that we think can fix Fragile X. So anyway, happy to take questions. Thanks for your attention.
Derek, thank you very much for introducing milestone research, which rewrote the textbook, as well as new findings, including unpublished data. So any questions? Yes.
Very interesting talk. Mechanistically, how do these accessory proteins regulate the channel, change the structure of the pore so that they become, calcium permeable and to different degrees?
No, yeah,
Yes,
it's a great question. So if you go into the paper in the last figure, we do some modeling, and what we show is that they must be changing the selectivity filter. And because calcium ions, you think of the size of the select filter, you might not have to change it more than an angstrom to get it. And that's very hard to catch that from a structural basis. You have to get the resolution way down. And so I actually have a colleague in Cambridge who's trying to do that right now. And I'm sort of saying good luck, because it may be very hard to see it. But what we showed from a modeling perspective, that you can actually get quite small changes, and that breaks calcium through. What's interesting is they don't like polyamine blocks. They're not blocked by polyamines. And that's a much bigger molecule that goes into the pore and gets blocked. So it's telling you it's about size, right? So very small changes in the pore dimensions is enough to make them exactly perfect for how our brains develop. And then for these kids, it's maybe just a little bit too much, right?
yeah, thanks.
Any other question? Oh, yes, please.
Thank you for really, really nice talk and research. I'm very surprised. And then my question is, so as you said, you think that the loss of spermine blocking could be a big contribution of this disease phenotype, or like a change in selectivity, also like ion conduction pattern?
I think so. So the GLUE2 lacking AMPA receptors, the Bergman glial cells, they're actually very strongly blocked by polyamine, and that was my kind of start in science, right? Where I kind of discovered that with Mark Mayer. Maybe I can get to it. I think the polyamines are actually, they're in astrocytes most of the time, and if I can find it, it'll take forever to get there. But I think the polyamines are actually here. I think they're actually here. I think they're actually restricting calcium transport. So the astrocytes don't get too much calcium, but they just get enough, right? Whereas magnesium block of NMD channels, it's the opposite, right? They shut down calcium at resting membrane potentials and only open when things get activated. So in that case, yes. But I think in the GLUE2 containing AMPA receptors that I just talked about, they're not blocked by calcium, but they don't transport calcium as well as the GLUE2 lacking ones. So they offer a kind of more modest increase in calcium. And there's a whole bunch of things that we haven't yet, we're kind of interested in looking at. We haven't explored fully how that might be, but you can imagine when synapses are formed. I know this audience isn't a big neuroscience audience, but when synapses are formed, there's this kind of battle of which synapse is going to stay and which synapse is not going to stay, right? And then all through life, we have to keep those things, right? And this little amount of calcium, remember, the real estate of calcium in neurons is very, very important, right? It's not like that in muscle or something, but in neurons, it's very important. And so it might be enough that the synaptic transmissions bring enough calcium in just to tell the synapse, yeah, just stay there. You have to stay there. And as we get older, as I'm feeling now, maybe that process doesn't work as well. We start to lose our synapses. But, you know, that could be one way to think about it. It's a homeostatic.
Okay, thank you. From the interest of time, I'd like to close and thank you very much for your presentation.